Um, so welcome everyone. It's a great pleasure to have Mahendra Rao here for this evening's public lecture on stem cells for therapy, hype, hope, and reality. In particular, because of the way stem cells have been bandied about, especially in our ancient Indian traditions. And hopefully we'll find out what contribution those traditions have made to our current understanding of stem cells. But I suspect we will hear a lot more about hope and reality when Mahendra comes to this podium. But let me tell you a little bit about Mahendra. He started his career as a medical doctor in Bombay. He got an MBBS from the University of Bombay, and then went off and did a PhD at Caltech in the Laboratory of Neuroscience, and very quickly began to work with David Anderson also as a postdoc. But he tells me while he was at Caltech, he was in fact surrounded by NCBS denizens, Matthew and Mani. Many of you who know Matthew and Mani were infecting Mahendra with the NCBS bug. And Mahendra, although he didn't spend as much time as he could at Caltech because he had to shuttle between Cleveland and the East Coast, I guess, where somewhere close to the East Coast and the West Coast of America, because his wife was a resident over there. Because had he spent more time with Mani and Matthew, you never know what could have happened. But then, he very quickly became a professor at the University of Utah, and he was one of the first people, one of the really pioneering people who started their own laboratories on trying to understand stem cells and stem cell mechanisms when he moved to the National Institutes of Aging at the NIH, in Bethesda. And this was, in 2005, where people were just beginning to think about stem cells as an entity that could be manipulated in the laboratory. He then, at that time, also realized the therapeutic potentials of stem cells, and started two companies. Well, not at the same time, and he continues to be the founder and the chief strategist of Q Therapeutics, and he has a couple of other companies that he has founded with things that he and his colleagues have developed in this area of stem cell biology. But Mahendra continued to be interested in pursuing sort of fundamental research in stem cell biology. He was invited to the NIH in 2011 to be lab chief and director of the Center for Regenerative Medicine at the NIH, where he developed a full-blown program at the NIH to explore the possibility of creating a whole repository and a repertoire of stem cell lines that would be available as a global resource. He, of course, went back and forth between academia and industry and has driven a number of various companies as well. He's sort of been a vice president of research in regenerative medicine at InVitroGen. And currently, after he will perhaps tell us the story better than I can, but after the bureaucracy in the government system got to him at the NIH, well, even at the NIH, he then escaped to New York, where he then helped the New York Stem Cell Foundation do the work that they do currently, which is really be a major funding center and a resource for stem cell biology. Well, as I said, we very early on in this trajectory, Mahendra intersected with NCBS, and we had, he had been enticed by Panex to come and do some work here, and we appointed him, and he, I'm happy to say, considers that as one of his honors, because if you look at his CV, it says that he's an adjunct professor at the NCBS, and he continues to be so. And given this long association, it is no surprise that Mahendra has come back to help build what I think is an extraordinarily exciting program on I guess, the developing stem cells as a global resource from an Indian context. And that's an incredibly exciting thing, and perhaps Mahendra might allude to this towards the end of this talk. I would like to welcome Mahendra. It's a great pleasure again to have him here.
Thank you, Satyajit, for those kind words. When my wife talks about my career, she says, you're the guy who can't hold a job, and that's why you had to move so much. So there's a difference in perspective, and that's what I'm going to try and tell you here as well about stem cells. But before I start, my association, it's really a pleasure to be here. And my association with NCBS actually was when I applied to NCBS for a job soon after I'd finished my PhD. I'd actually gone to do my PhD because as a neurologist, when you try and treat patients, you can make a diagnosis, and there's not much you can do after you made that diagnosis. So I wanted to do some research in neurobiology, and that's why I went and did a PhD. And then I thought I'd come back, and everything was set. Obaid Siddiqui actually interviewed me, and I said, yes, I'm ready to come. And that was just the time when NCBS decided that they were going to move to Bangalore. So I was all prepared to go to Bombay, and then we were going to move to Bangalore, and so it became a little bit difficult for me because my wife had already looked at positions there. And so that changed my career. Because I hadn't applied for a postdoc or done anything at that time, and so I had to scramble to find a job. But that changed how I was doing things. So I've been working on stem cells well before stem cells became fashionable. And the reason I was working on stem cells was simply because we were interested in how the nervous system develops. And for the nervous system to develop, you have to go back and look at when the cells are actually born, and you have to go back earlier and earlier in early development, and then you realize that only dividing cells that are present early in embryonic development. And so that's how I was led to stem cells and trying to understand how they make fate choices. So what I'm going to try and tell you today is a little bit of background on how the world views stem cells, and then tell you about what the reality is with what we can do today. And it's not as exciting as what people make it out to be. But I also want to leave you with the idea that there's a lot of potential here as long as we do the right things. Okay? So here's a brief history of stem cells in terms of how people have considered this for regenerative medicine or replacement or repair in the body. It all started with the fact that in hospitals you could identify and harvest tissue. A lot of it was just surgical waste. And then a lot of clinicians said, we should be able to use it. It's healthy living tissue, which we had to take out for one reason or the other. And so people used to harvest what was called biologics from either donated samples from patients or from cadavers. And then this was processed in some fashion, and you used it. So it could be dried bone paste, or it could be tendons which had been taken out, or corneas which were used. And all of that became a business, right? So hospitals would get that. Companies would process it, and then this would be sold and distributed, and that's how a large number of patients were treated. So keep that in mind. So that was how the whole history of processing biologics started, right? And as people were doing this, there was a lot of excitement, and this was actually done in the United States first where people thought that you could also transplant living cells. And the first thing they looked at was doing bone marrow, because skin grafts had been done before. You could mobilize skin from one place to the other. And so the question then became, why can't we take cells from one person and do it in another person? And the easiest cells to take were circulating cells, where you didn't have to really cut up anything or chop it or harvest it, and bone marrow transplants were done. And the first successful bone marrow transplant was done well over 50 years ago, and it was done by twins, and everything worked. So the first one worked, the next 10 didn't work, and all patients died, right? And that's when they learned that we have to do something more than just take cells and just put them into anybody. And they realized that there's an immune system, and that you have rejection phenomena, and you have to learn all about rejection and matching. Soon after you could do that for blood, and you got some development there, and there was a huge, big breakthrough that occurred in terms of first learning about the HLA type, and then learning about immune suppression, and identifying cyclophosphamide. So that was a big breakthrough in what we could do. And soon after we could do this successfully for bone marrow transplants, people started doing organ transplants as well. Then about 25 years ago, people said, we are not getting enough bone marrow cells. And there were these international registries that were set up, and you have donor registries, and maybe some of you have also opted to be donors for bone marrow. Though in India, the number of people who opt is quite small, they looked to see whether there was an alternative. And somebody had this bright idea. It's a person by the name of Elizabeth Catino and Hal Broxmeyer, who thought about cord blood. It's placenta. You can milk the placenta, and you can collect cord blood. It has the same kind of stem cells that are present in bone marrow. And you could then see whether you could transplant that. And the story is quite amazing, because the first cord blood transplant was done by shipping cells to Italy from the US. And the rules were very straightforward there. You just decided you were going to do it. And the reason I'm telling you this is it's important, because some doctors feel that they can still do it that way. And the story that's told, and it's actually true, Joanne Kurtzberg, Hal Broxmeyer, and several of the others are still active physicians and are working, is that somebody actually dropped the cord before they the cord sample, the blood sample, and they just picked it up, and then they did transplant it. And luckily it worked. And I actually met the individual in whom it's transplanted. He's healthy. He's actually got children of his own, and he came to the Duke Cord Blood Center for a photograph with the physicians who worked with him in that. So at that stage, then we had bone marrow, we had cord blood, we knew how to do organ transplants, and we had all of these immune suppressive drugs that we could use to be able to do things. There was another big breakthrough after that, was then people said, what is present in bone marrow, etc., that we can use for transplants? And it turned out that they were able to identify a subpopulation. It was called the stem cells, and they called them hematopoietic stem cells, and those were the first stem cells to be discovered. Later, and this now I'm bringing you forward to about only 15 years ago, in 1997 or so, embryonic stem cells were harvested from humans. We had known about mouse embryonic stem cells, but now we could make embryonic stem cells from humans, and we could now show that they work much like what had been done with mice, and that you could make every stem cell population from that. And then, as you know, just in 2007, 2006, 2007, Shinya Yamanaka showed that you could take an adult cell and make something which looked like an embryonic stem cell. So now we knew that we had stem cells, and then there were a lot of breakthroughs and a lot of research papers which showed that we could take these cells and we could differentiate them into appropriate differentiated cells that you could then use and transplant. So now we can take embryonic stem cells or induced pluripotent cells and make liver cells or cardiac cells. You can make intestinal cells, you can make brain cells. So you can make pretty much, let's say there are 212 different types of cells that have been identified in the body, and you can make maybe 70% of them. Okay. One more breakthrough, and I'm going to just end with that in the end, was that normally we can make cells in culture and you can do 2D cultures, right? But recently what people have shown is that you can make organoids. So they look like mini organs if you can do the culture appropriately. And that's been a really big advance, because we know that interaction among cells is very important as it develops. And then there have been really, in the last 10 years or so, really major advances in gene therapy and in bioengineering and in large-scale data analysis. So keep all of this in mind, because this gives you the background of what's been happening with stem cells in terms of what one can do. So when you think about this, the first thing is it has to be all translated from research into practice, and the people who normally do this are companies. So in the next few slides, I'm going to just go through a very brief outline of what kind of companies and what happened, right? So this is a brief overview of the field then. You've got tissues, scaffolds, devices, and organs, mostly dead cells, and there are companies which do all of that. And then you have living cells, which you can put in 2D structures or 3D structures. You can combine it with certain things, and you can have completely artificial substrates. And on the right-hand side, what you see are some examples of all of these. So at one extreme, you could have something like a retina chip, which has almost no cells in it other than the connections that you make at the end. And you can have 2D structures like skin and valves and cartilage, which are all dead cells, which are processed from cadaveric tissue, all the way down to a mixture of these things. And then if you look at cells, just like I started by telling you the history, probably the largest amount of the work that's being done is being done with hematopoietic stem cells of some kind. Then it's cord blood and ancillary tissue. They later found an additional type of cell which was present in the bone marrow, which was called a mesenchymal stromal cell or a mesenchymal stem cell. And then later there are other stem cell populations like the neural stem cells and liver stem cells and intestinal stem cells. And I mentioned neural stem cells here simply because that's the one I'm going to tell you a little bit about. And of course, when you think about therapy, it's important to remember that when you think about therapy, it's going to be autologous cells where you don't have to worry about immune suppression, or it'll be allogenic cells where you'll have to match and you'll still have to do immune suppression. And you might engineer them, or you might do it in combination. And those are all the different kinds of cells that people have already begun using. Okay. So the first set of companies were things that did organs and tissues and so on, which were processed from dead cells, right? And these are all companies that were established in being able to do that. It doesn't matter what they are. The important thing to show you is that it's a big list, and there were companies which were established in most parts of the world, including in India, which provided either skin or placental membranes or cornea, etc., to different groups so that they could use it, and probably orthopedic applications were the largest of these sorts. Right? The next big thing that happened was really you could already take cadaveric samples of valves, etc. So people said, well, why don't we just take that as a scaffold? So it's a natural scaffold, and add some living cells to it, and maybe that will increase the utility of the cells. And that was what was done. And I'm just showing you 3 examples of something that's more extreme. This is a bladder which has been made from cells from that same individual. You harvest the cells, you put it on a scaffold, and you grow a bladder in a dish. It's pretty thin because you can't make it too thick because you don't have vasculature, but this is good enough that you can actually implant it, and then it will function and it will have sphincteric function as it develops, and this has been done in several hundred patients. There are people who have cancer surgery and then they lose their trachea because you have to operate around it, and then you can replace that so that you have things with lungs that you can then have air connection, and then that has worked and several people have survived. One of these things was done in Baltimore at a hospital where I worked, and so it's kind of nice to see that. Unfortunately, the patient died for other reasons after 2 years after having this artificial trachea. Valves have been done for a long period of time, and what you're seeing there are blood vessels. Okay, so that became the next sort of evolution. So we're just doing an incremental thing about taking something and putting it there. So that's the reality, right? This is all being done routinely. This is being done at different centers. Each of these things has been used to treat patients, and patients have been successfully have survived, let's say, these experiments. Okay. So the next thing, of course, is when you do all of this is that you start thinking about how you can use them in other creative ways. And there are lots of trials, and I won't go through all of that, except I want to highlight one simple thing here, and that's this number this is old, so this is about 2 years old now, but there are 33 registered trials in the NIH clinicaltrials.gov registry. It's a voluntary registry, but it's telling you that there's activity that was happening in India as well. And this was legitimate activity in terms of what could be done. Okay. And obviously, just like there were companies which were doing dead things and products that were there were also companies which were set up to try and make products out of living cells. And I just want to put some out here just to point out that they were doing it with different cells, and they had different names, and they were present all over the world. And there are companies that are present in India as well. And these are just examples of different companies. Okay. So that's sort of where the background history was. So I've tried to bring you in a very quick way, fast forward to where we are today. Is there are certain things that are already being done. There are dead products that are there. There are these biological products that are available, and patients are being treated. And I'll just give you one number before I move on to the hype part of this. Was with bone marrow transplants in the United States, about 30,000 people are being treated every year. Okay. With cord blood, it's about 5,000 people that are being treated every year. So that's sort of the relative number. With all of the others, it's much smaller, but that's sort of the extent of what's the activity which is legitimate in the field right now. Okay. So what's the hype? So the thing here was that surgeons and physicians had been doing all of this, and they'd taken cells, and they treated several thousands of patients, right? So, and the assumption was, well, stem cells are stem cells. So, stem cells are somehow magical, right? And what people lost during that discussion was that all stem cells are not the same. So the assumption that this was this one big error that was there in the whole field, and we scientists are partly to blame for that, right? So people said, well, we've got a bone marrow stem cell, we've got a mesenchymal stem cell, but they only heard the word stem cell, right? And so they said stem cells can be used, and they're everywhere in the body, so somehow they can treat things, right? And the other assumption that people made was, well, if the stem cells are everywhere and then they can do these things, they must be smart because they know where to go, what to do, and they do the right thing when they get there, right? So that was an important thing. So they'll respond to injury, they'll be able to do the right thing. And some people even went to the extent of saying stem cells are magical, that they're not identified as foreign by the immune system, so the immune system is not a barrier, right? So all stem cells are the same. They know where to go, they know what to do, the immune system is not a barrier. So what does that mean? It means a physician could take cells, identify stem cells that were present, and if it was easy to do, just do it like any other surgery and put them back in the body and use it to treat anything, right? So the same drug or a pill, you don't think that you can use it to treat everything, but then the people said, oh, stem cells, they can, because they'll change and they'll recognize what can be done and we'll be able to do this. So the next thing, of course, is whenever there's new technology, there's always a barrier because, either you need like a big instrument to do something or you need some specialized facilities to do this. But this was fairly easy, right? They'd been doing bone marrow aspirations for bone marrow transplants anyway. So heck, anybody could do it, right? And then if you're harvesting fat, it's very easy to do. So it meant that you could do this sort of thing without needing any expensive equipment or having to do a lot of processing. So anybody could do it, right? So that's what caused this whole explosion, right, of people making claims with things that could be done, which there was no scientific data for it. And the other thing that people forgot, which I didn't add here, but I wanted to make sure I emphasize, is scientists, when they were doing research, had discovered what they had called the placebo effect, right? And the placebo effect is quite strong. And for those of you who don't know, it's something like this. It's, when your mother, when you're a child and you go running to your mother with something that's wrong, she says, I'll kiss you and it'll make it better. And suddenly you feel better and you see all pictures or things about how that happens. It's the faith that something is going to work, which is sufficient for you to feel better about it or make your body change, right? And that's a really important thing to keep in mind. And that's people could not quantify it, but people did clinical trials and studies, and they found that this was extremely strong, and often it was stronger than the effect of the drug itself. And if you didn't take it into account, you would have huge problems in how you interpreted results. And so if you did something in a small number, and you had patients who were desperate, and it didn't matter what you did, right? Some people would get better, and then you would say, see, it worked. And so that's what happened. So you had a lot of stories about people getting stem cell treatment, and that they got better, and then people had this hope because they didn't have any other choice, and then that would work. And the reason why I'm emphasizing this is that when you took these preliminary results, which looked all exciting, and then when you did a rigorous sort of blinded study where you could eliminate the placebo effect, then many times all of these magical results went away, okay? So keep that in mind as well. But physicians don't do clinical trials, right? They treat one patient and then they send them home. So they were not going to be able to dissect out the result from a placebo effect as well, okay? So the hype became, look, we can take these cells, all stem cells are the same, so it doesn't matter which stem cell we use, and we're going to be able to treat any disease because they'll be able to go anywhere, so we don't even have to put them in the site where it's damaged. We'll just put it in the blood, and then we'll send the patient home and they'll get better, right? And some of them obviously did get better, right? So, and that became what I call a suspension of disbelief. And we tried bone marrow and mesenchymal cells for every indication. We forgot that the placebo effect exists, and we ignored the fact that there might be risks of putting cells which in the wrong place or the wrong time or in the wrong numbers to be able to do it. And we also ignored the fact that when you culture cells, it might change their character, and that became an important issue. And the other thing we forgot, and this is like in homeopathy, right? When you think about a really small amount of something can work, but small numbers of cells, if you transplant in a big, organ, they have to fight whatever is happening in a really small number, whether that work or not work. This didn't stop people, and many, many people did it. So hundreds of trials, thousands of case studies, several deaths, and many arrests for breaking the law because of processing things or making claims which were not adequately supported by the data, right? And that's sort of the current status, despite a lot of effort that's been made to do this. And here there was a big problem for any of the countries in which all of this was being done. Nobody had thought about stem cells and how people would use them. So there weren't very clear-cut rules about what is prescribing a drug and what studies you have to do before that versus what is practice of medicine and what doctors can do on their own. So that was a big problem as well. So in many countries, you could say, oh, all of this is bad, but this is legal, right? And so you're not breaking the law in being able to do that. In other countries, people scramble to start changing the law. But it's not clear that they changed the law the right way. So we had issues on all of those fronts, right? And India too, this was true. So India is not immune from all of this as well. And we have many other clinics that were set up here. There are people who sell cells with wild claims on what can be done. There are clinics which advertise the use of stem cells for any indication. So I can come to India and I can get cells transplanted for spinal cord injury. I can get it done for stroke. I can have it done in my heart. I can do it for cancer. And there are people who are willing to do all of this. And I just took two, and it's not like to highlight anybody, but I want to emphasize this just for one important thing. There's one cell type which is different from all others in that the embryonic stem cell has one added risk. And that added risk is not that it's somehow bad or worse. It's just that that's its potential. It can make every cell type in the body. So if you transplant that cell and it doesn't have the right signal to do what it should, then it'll just grow, right? And what you get is a benign mass, which people call a teratoma. And then within the mass, there'll be random differentiation. And people assume all stem cells are the same. So they said if we can do all of this with bone marrow and we can do it with mesenchymal cells, we should be able to transplant embryonic stem cells as well. And that's what Gita Shroff was doing. And there are some other groups in Russia, etc., who did this. But the problem here is you put too many and they don't differentiate, then you're going to get a mass. And if it's in the wrong place, that's going to kill the person. And that's what I said about in some places there were several deaths that occurred after this sort of stem cell therapy, and that's what made it important that it could be done. So that's the hype. Every country that I have visited, there are many clinics. Many of them, you can go to their websites and they will advertise services, and they'll tell you we can put a stem cell in and that we've cured many patients. And look, you can even get a testimonial, but nobody does a clinical trial or clinical study to be able to prove that. Okay. The other place where there's something where you say, well, if it's against the law, how can we still make money in being able to do that? And that's the field of which is sort of the borderline between actual therapy versus claims that you make in the cosmeceutical market, right? So if it's a drug, and you can use it to treat acne, but you can also say it's not really a drug, and you can have something that's a cosmetic which can be used to treat skin disorders, for example, right? And so this is just on the flight when I was coming in the flight magazine, I saw this, so I just took a picture of this and said that, so you're seeing people selling all of this. Korea is a really big market for these sorts of cosmeceuticals which are done, but no big company is immune from it. So if you look at L'Oreal, they advertise extract made from plant stem cells, which is anti-aging, right? So in being able to do that. So this is another big market, right? And I remind people when I talk about this that it's like the internet, right? When the internet started, it was a university and a defense-based initiative, and the idea was that it would be a service for research and you could share data independently. But guess what was the first use of the internet, right? Anybody? I'll tell you, it was to sell pornography and for gambling, right? And that's how they made money. So it seems like that's the same thing that's happening with stem cells, is they look at the opportunity to make money and say, it's not really curing patients, but it's being able to sell cosmetics. Okay. And medical tourism became a big business, and this is true everywhere. And the reason I said this is that the placebo effect and the conviction that this works claims everybody. And in Texas, for example, the Governor Perry actually went to a clinic which was harvesting cells and putting them back to treat all sorts of things. And it was against the law, but he advertised that he had done this. And because it was so important and he felt better, he wanted to change the rules and abolish the FDA, which was telling this company that it was against the law. So you can see that there's conviction that has to be there about what is legal and what's appropriate and what's not, but certainly that's the hype that's out there, and that's what's happened. So I actually visited several of these clinics as part of a fact-finding mission, etc., to be able to do it. And it's amazing how much money they charge and how much gets done. And the depressing part of all of this in my mind is that there may be some benefit, right? But we just don't know, right? And if we don't study it carefully, we won't know how to make it better, right? If you've got a 10% benefit, it could be 90%, right? And we just will never find out if we don't do it properly. So that's the hype that I want to tell you about. And then I'm going to tell you a little bit about the hope with stem cells and move that forward. So as this was happening, there were rules about doing this, and people started trying to say, well, let's try to do this properly. Let's think about the placebo effect. Let's try to make sure that all of this can be done. And again, I'm not going to discuss any of this in detail. I just want to tell you, so there's a legal group of people who are trying to move things forward. The important thing to remember is that average time to get a drug to a market is about 8 to 10 years. Cells are at least as complicated as small molecules, so it's going to take the same amount of time, and it's going to cost the same amount of money as it takes to take a small molecule to the market, maybe more, depending on what indication you're looking at, because your manufacturing costs are going to be more. Nevertheless, there are several companies which have begun this process, and they are in phase 1 or phase 2 trials, and that's what you see listed on the right-hand side here. And they are looking at a variety of diseases to be able to get that done. And the reason I show this is that these are companies which are publicly traded. That means they must have gone through angel investors and people who might have just put money into them on faith, and now these are guys who are thinking that there might be actually a profit or there's some substance here to be able to do it. Doesn't mean it'll all work or that's all going to be successful to be able to do that. And that was mesenchymal stem cell companies, and these are just cord blood companies. So you can imagine with each stem cell type, there are people who are trying to move things forward in being able to do it. So then companies have got products to the market. So that means they're now actually legally approved and they're being sold, or they're in the late stage approval process. And so this is just a list from the United States of things that have been approved. So you can have a device which processes cells which can be approved. You can have combination of gene and cell therapy that can be approved. There could be different cells that may be approved. So here these are either approved, pending, or late stage process. So for example, there's one gene therapy that's just been approved in Europe. There's some skin product that's been approved from one of these companies, and there are others which are in late stage approval processes. But the actual number of approved products in the United States is quite small, and it's the second largest after Korea. And in most other countries, it's smaller. Okay, so that's the reality in terms of being able to look at it, or the hope that these will show large amount of effect, and then we'll be able to go forward with that. So there are some cord blood products. There's a fibroblast product. There's a skin product. There are several other skin products, and there's a device. So a total of about 9 products, of which 5 or 7 are true cell type products, right, that have been approved. And there are about 14 such products which are in late stage phase 2 or phase 3 clinical trials. So if that's what's there in the United States, what's happening in India? So in India too, if you look at what I just told you about this whole history, Reliance probably is the most active in sort of the dead cell products and using one specific type of stem cell in this country, which is the limbal stem cell for cornea, and using things like placental extract, etc., that is being done. Stempeutics is a company here based in Bangalore, which has a mesenchymal stem cell-like product, which is for critical limb ischemia. Shree Chitra has some scaffolds and synthetic material that's being used to treat patients, which was all developed here. And the government is evaluating changing rules for how products may be approved, because there was no formal approval process in India for cell therapy products that could be done. And it wasn't even clear whether the FDA or DGCI would approve it, or whether it would be done through ICMR. Now there's some clarity on what can be done. However, India has a problem or a limitation how it can move forward in this arena. And part of that limitation is we don't have a large number of bone marrow transplant centers which can do large numbers of bone marrow transplants. And we don't really do completely HLA-typed transplants because we don't have access to that many. There are a few centers which are doing that. Maybe it's CMC Vellore is one big center that does that. AIMS does transplants, and Fortis is a private hospital, does transplants. There are several cord blood banks, but there are only private cord blood banks. And private cord blood banks, the use is going to be quite limited because not all the people who store their blood are going to need it for bone marrow transplant purposes. So there's no public cord blood bank. There's one public cord blood bank in India that I'm aware of. That's based out of Chennai, but the number of units that are stored in that is very small. So there's a limitation of even having access to cells that you can use to test for various indications for being able to do it. There are several centers for stem cell research. Yes, InStem is one of them. And there's a recent approval for allowing cell products that can be imported into India so that you can do, have treatments. And so there's several such products which are now becoming available in India. Okay. So this is the part when I said about looking at reality and sort of hope that these are the things which will move forward, is that we know that the field is maturing when companies go out of business as well, right? Because it says they tried something, people tested it, and they didn't like it, and then that failed. And I want to just give you three sort of small stories. These are MSc companies which have now gone out of business because of what they were doing. So Provenge was a company which had NK cell, a natural killer cell therapy, and that was actually approved. But the approval was because it allowed to, it was for treatment of cancer, and it increased lifespan by about 4 months for terminal stage patients. And the cost of that therapy was about $100,000. Okay? So this is important to remember. So what happened was physicians looked at it and said, well, it's great, we can do this, and it works, and the FDA has approved it, but it's not of enough benefit for us to recommend this to all patients, right? And so they expected to be able to sell a lot more than they could, and their plans were such that they had to file bankruptcy. So the company had a good idea, but because of planning, they could not get that work. The same thing happened with skin products. There were 4 companies. Each one of them was actually selling a product that was useful, and their sales were in the range of between $100 to $300 million a year, right? But their costs were more than what they were making on their sales because their manufacturing was not done in an adequate way. And so all 3 of those companies went bankrupt as well. So that's the important learning to be able to say that. This is the story of Geron, which did embryonic stem cells. They spent a lot of money. The amount of money was so much that they decided they were going to abandon it without even making a final decision on whether the product was useful or not. So what this tells you is the reality is that there are many efforts moving products forward for appropriate use under appropriate regulations. But if you don't take into account not just the science, but the practical issues of the cost and the benefit of that therapy, then probably you could suffer the same fate. So it's an important thing to combine what you're thinking about doing on the research side with what is the applied work that you might need to do as well. Okay. So this sort of gives you a whole really brief outline of where we are, right? So we have many companies, many things moving forward under legal programs. There's a little bit of illegal work that's still going on in clinics, which is unregulated, or it's legal in some countries, and so there's medical tourism where people go for that sort of therapy. And there are several products which are potentially exciting, which are in late stage phase 2 and phase 3 clinical trials. In the next few years, we'll know about how good or bad they are. So I'm going to just tell you a little bit and highlight some of the things based on my own efforts to try to follow this appropriate path and do that. And again, the importance is not what exactly we are doing, but as related to the issues that I just brought up here, right? So we work in the nervous system, and as you can imagine, the nervous system, everybody's trying to do a whole lot of things here, and they're trying to treat a whole lot of diseases. The one thing we learned from all the people who had been doing this so far was when they rushed to put mesenchymal cells or other cells back into the brain, that they didn't really work. Okay, so for a chronic disease, if you put a cell which was not normally present in the brain, it didn't seem to help, and if you just put it through the blood system, not many cells went into the brain. So now there is a new set of companies which are looking at using neural cells, and the neural cell choices are sort of limited, and in the brain you have sort of even fewer choices, because it turns out that if you put neurons in one place in the brain, they don't migrate or go anywhere in being able to do it. And if you put oligodendrocytes, they only make myelin, so you can use it to treat disorders where there's a myelin defect, but you can't use it to treat other disorders. Astrocytes provide trophic support, but they don't replace the neurons. So what you can do is limited in the kind of therapy you can do, but you can reduce the amount of damage in a stroke, for example, if you use the right kind of cell. And so people are trying to use these sorts of cells, put them back in the brain in a local or focal type of disease so that they can then use this for therapy. So we looked at all of this and said, great, what that means is we need to make astrocytes or glial cells or glial precursors which can make astrocytes for therapy. Or if we make neurons, we have to make neurons which will be in one place, and the requirement will be only in one place. And one example of that is in Parkinson's disease, where the neurons are lost in only one site, and so you could replace cells in that site, or in retinal pigment epithelium, where it's like the support cell which you can provide for the degenerating rods and cones, and maybe that will prolong the life of the rods and cones that you can do. So again, you can see we are now limiting the hope, right? We are saying that we are not going to cure the disease, but we are going to retard the progression of the disease. It's a very important distinction, but that's somehow what was the reality and what we can do. So I won't show you all the data, but this is where we are. So we started all of this work maybe 10, 12 years ago. That's how much time it takes to be able to do this. We have an approved clinical trial for treating motor neuron disease with fetal glial cells. And we have a next generation product which will come after this with making the same glial precursor cell, but now making it from iPSC, so we can solve the immune system problem as well. This is being done in a company called Q Therapeutics, and the company, and I'm just going to give you these numbers because it's important, has already spent more than $20 million to get to this stage, and we have yet to go through phase 2 and phase 3 to be able to do it. So that's how much time it takes to be able to do it. We have a second product which is making astrocytes, both from fetal tissue and from iPSC, because astrocytes may be better for certain kinds of diseases than the precursor. And we can engineer the astrocytes using these modern gene engineering techniques like zinc finger nucleases, talons, and so on, so that you can make engineered astrocytes. We also can make dopaminergic neurons as a product from both stem cells and induced pluripotent cells, and we can make a neural stem cell, which some other groups are going forward. We decided that we don't want to use this, so it's available for license. Okay, so all of this is what we can do, and what it means is you have to manufacture these cells and be able to get that done. And so the next few slides, I'm just going to rush you through. It doesn't matter in terms of the details on how we were trying to do this. So the first thing, because the paper has been published, we were able to make clinical grade iPSC lines, right? And we could take these lines and we could use them to be able to manufacture all products. And this was important just for one reason, is if you do this right, then what you can do is you can start with pluripotent cells, and you can make an intermediate, which is a neural stem cell. This cell can still expand, and that's how it grows over a long time period. So this intermediate shortens the time at which you can make the more differentiated cells, and more importantly, it gives you consistency when you manufacture from time to time. And because you're using the same starting material to make multiple products, it reduces the cost. Okay. So remember, I told you that cost is going to be a very important thing as you want to move these things forward. And so we've shown that this can all be done. We've shown that you can manufacture pluripotent cells, you can make these NSC, you can make these astrocytes, you can make this glial precursor, and you can make dopaminergic neurons. Okay. We can make multiple other neurons, and I'll skip that. The important thing is that that can all be done. So we have a modular manufacturing process, right? We can identify the pluripotent cell you want to use. We can see if we can make an NSC bank. We can then expand and differentiate into the final product in a cGMP facility. We can take that product and provide preclinical safety data. We can then file an IND to do this, obtain the appropriate safety data, get marketing approval from the regulatory authorities, and then once you've got the final data on a phase 3 trial, you can get final marketing approval. And because we can treat orphan diseases, we can accelerate the timeline, so this can be done. But this whole process is still going to take 5 to 8 years to be able to get that done. So it costs money and time. And for example, one reason Q Therapeutics can't move forward right now is it doesn't have enough money to do a phase 2 or a phase 3 trial. So that's sort of the reality of what can be done. Okay. So that's the reality for many of the companies which are moving these things forward. I just gave you an example of the nervous system from our own efforts, but there are about maybe 20 groups doing the same sort of thing, and likewise there are other groups doing this in the cardiac system or in the liver system, etc. Okay. So I don't want to make it sound very pessimistic, right? I mean, what I've said so far is, here's somebody telling you that we can cure things, and then I'm telling you it's going to take me 8 years just to retard the progression of a particular disease, right, in being able to get that done. So what do we need to be able to make sure that things happen even better? Or is there hope in the horizon, and what kind of breakthroughs will you need to be able to get that done? So I'm going to tell you about some of the things that people are trying to do. So one thing that people realized was that because you can get multiple sources of cells from pluripotent stem cells, is that you don't have to use a single cell for therapy. You can do combination, and you can make organoids, and you can make 3D cultures to be able to do that. So it gives you a really whole new paradigm for treatment. And because you can do this in a personalized way by doing HLA mapping, then you might be able to make a haplobank to be able to get that work done. there are these novel engineering methods, so you can do gene editing and you can do safe harbor gene therapy, etc. You can do that. And then what people realized was that we don't need to go all the way back to a pluripotent stem cell stage. If we have all of these engineering techniques, we can do what people call transdifferentiation. So you go only partly back, right? So you take an astrocyte, make it go back to maybe like a neural stem cell so that now you can make neurons from them, rather than going all the way to a pluripotent cell stage. So that allows you to make things much faster. And you can do it from the same person, and you can maybe take adult cells and do it without having to collect a clone to do it. So this can give you differentiation. Then people said, why don't we do direct differentiation like this, but don't have to put it in culture at all? Why don't we just do it directly in the brain or directly in the heart? And there are people who are doing that, and there are studies that are ongoing for that. Deepak Srivastava, who's actually visited here at one time, is doing some of that work at the Gladstone Institute at UCSF, for example, looking at the heart to be able to do it. The other exciting thing has happened in this sort of whole thing about making 3D cultures and organoids and introducing vasculature into those tissues. And there's been some work which has been done at MIT which allows you to do that. So now you can see that we could truly imagine not just making a mini organ like we can do with organoids, but you can actually try and start thinking about making a complete tissue. And there was this one big excitement on something called decellularization and recellularization. So what you can do is you can take a scaffold, essentially a living kidney or a liver, and which has been donated from a cadaveric sample, and you can kill all the cells within it. But now you've got this whole scaffold, and if you do it right, you can then pump it with the cells you want, right? And when you do that, you actually get a whole organ. And it's pretty dramatic, and I didn't want to show you that a video or anything like this, but you can take a heart from a mouse, for example, and you can kill it, you can kill out all the cells and repopulate it with cells, and you can show that the heart can start beating again, right? So it makes a beautiful picture when you're looking at that. So that's called DeCell ReCell or decellularization, recellularization, and that might allow you to make organs in a very quick way. There's another company, for example, which is saying we can make organs, we can take advantage of biological animals, and we can grow organs in an animal just and then use that. So we can humanize, or we can transplant the right kind of cells so that we can do this kind of DeCell or ReCell population in a living organism and then be able to harvest that organ for replacement. And that's work that's being done in pigs that people have thought that they can use. Okay. Are these the only problems that you have to solve? No, there's one other important problem that I didn't talk about when I talked about all of this cell therapy, is that you have to get the cells delivered in the right place and the right time as well. And so one put, in each place people are thinking of new or experimental ways that this can be done. And it turns out that there have been some interesting things that people have found. It turns out that in the brain you might be able to deliver by the intranasal route. In the lung you can actually do by inhaling cells and being able to deliver it that way. And so there's lots of different sort of things that people are trying to be able to do that. So that's potentially quite exciting, because if you solve that one problem, then you can certainly do a whole lot more than you could have done even recently. But I want to tell you one last thing about all of these things in terms of using cells. Now, I started off by saying that maybe the most obvious thing to use on all of this, or what there's been the most excitement about, is using cells for therapy. But using cells for therapy is a little bit broader than just taking cells or making a tissue an organ and putting it back, right? If you could use the same cells to be able to make a model system where we could discover a new small molecule drug, which you couldn't otherwise discover, then that would still be using cells to get therapy, right? And so people have started looking at those sorts of things as well. How can we use stem cells not necessarily to make a tissue or organ, but to use it to discover things which would help provide additional therapy for patients? And I'm going to give you some examples from this without going through a lot of the data, but I'll tell you what's been quite exciting here, and this is something I think is not happening in India, and it'd be really great if that could be done. So one is this idea of a tox screen. So all small molecule drugs are tested in rodents or tested in 2 species, and you test for toxicity there. But while men can be rats, all rats are not like humans, and so we have to worry about whether we are really getting the right interpretation of those results, right? So people did the test. They asked, there were several drugs which had been withdrawn from the market because after they'd been approved from the animal studies, they found that there was an adverse event in humans, and so they had to withdraw those drugs. So a simple question we could ask was, what if we tested them on iPSC-derived cardiomyocytes could we detect these toxicity effects before we introduced them in the market and before people died? And the answer was 6 out of the 7 in a blind study of the drugs which had been withdrawn in the market, which had passed and had been already approved for sale, and could be detected as having a potential tox effect on the cardiomyocyte. So they never have been introduced if you could do that as a screen. And so now in the US, the FDA is now requiring that all new drugs be tested on human cells before they are released for toxicity. And people have taken this one step further, because adverse events don't happen with everybody, they happen with a subset of people. So you can now ask what's unique about those subset of people as to why they have tox, and can we identify and not give the drug to them. And that turned out to be something which Joe Wu, who was at Stanford, did and showed very elegantly, that there's one of these cardiac drugs which has a sudden death syndrome, but it only happens in a subset of patients which have a mutation in a single gene, okay? And that it's a single channel, and you can screen for that channel. So that drug could be reintroduced with a black box warning that people who have this kind of gene SNP can't take this drug, right? And so now you've got something which is of benefit for people back into the market where otherwise you could not, because you have that risk. So tox screening has become a very exciting field in terms of being able to do it. And we, for example, have looked at neurotox, and other people have looked at hepatotox for being able to do it. Can identify causes of diseases. And the obvious thing is to say, well, we can take a disease patient line, and we can now look at that, and we can screen it against a control. And that's a little bit like what DBNS here is trying to do with the neuropsychiatric disorders. And that's important that we can do that. But it's also important that we can do it on a large scale, right? So we can now use large-scale data analysis in a way that we could not do before, and we can see whether we can identify it. And I won't go through all the data, but for example, my lab looked at this for Parkinson's patients. And we looked at Parkinson's mutations, and we were able to identify a pathway and what was a disease-causing pathway in that thing, and actually screen for drugs to identify novel candidates which could protect in that particular class of Parkinson's disease. We did the same thing with name and pick, and we were able to identify a class of drugs which could be used for name and pick. And in fact, one of those is moving forward therapy. Now we can develop models that were not possible before. Blood-brain barrier models, which you couldn't do before, retinal pigment epithelium in an organoid or a sheet, which we could not do before, or cardiac sheets, which we could not test before. And we can look at drug repurposing and personalized medicine, and that's already being done for cancer, where you actually take the cells or the stem cells out from the patient and a tumor, for example, and then screen with the cocktails of approved drugs to see which cocktail is best for that particular patient. And I don't want to show you this, but Dr. Topol, for example, when he does that, or the Mayo Clinic does this on a routine basis for no option patients, and they have rescued 10% of patients who would otherwise have died because they had no other option. These are no option patients. That's, while 10% sounds like a small number, it's pretty dramatic for those 10%, right? Because they had no other choice in being able to do that. But more importantly, we have got novel assays to be able to discover drugs. And I want to give you one example, and this is work that one would not have expected, but infectious diseases. We haven't had a model for certain infectious diseases for human disease. Whether it's been HIV in the brain, or it's been encephalitis of a particular kind in sensory neurons, where we never had sensory neurons which you grow in culture, or it's been leprosy, where we haven't had a human model, so we don't know how it really propagates, or for malaria in the liver, for the cycle in the liver. And now that we have these cells, it's been now possible to show that we can use them, and that we can discover drugs or the mechanism of action for these things. And it's amazing how we found novel things that have happened. So for example, in a particular kind of HIV encephalopathy that we see, it's only the astrocytes which are affected. They're the reservoir for the disease. It's not the neurons. So you can see how we can learn a whole lot of things by being able to do that. So this is quite easy to do. It takes a lot less time than the 8 to 10 years to do this. And so there's a lot of groups working on this sort of use of cells, and it's been quite exciting because there are already results, just like I told you with the cardiac cells in terms of the tox screen. Okay. I'll skip these two slides. I just want to show you 2 slides from 2 different groups at the NIH, because both of them have visited here and have presented, so I want to remind people about that. One of them is making retinal pigment epithelium from eyes, from iPSC cells, and this is from Dr. Kapil Bharti's lab at the National Eye Institute. He actually trained at CCMB, I think, before he went there, and now he is a lab investigator there. And what they developed was a way to make retinal pigment epithelium sheets. And those sheets can be used both for looking at screening for drugs as well as by actual implantation. And Masayo Takahashi was the first one in Japan who did this work, but this group will probably be the second to be able to do this sort of iPSC-derived RPE for macular degeneration. Okay. This is Dr. Swarup's lab, who's done screening, but not the typical screening that I just told you about. He developed this whole protocol where they could make reporter lines, insert them so that they could look at specific lineages, and then grow them in organoid culture so that you could look at late development. And so this is CRL, which makes the fate choice between rods and cones and occurs relatively late in development. But now he could take these organoids and he could now screen to understand what was the regulators that were there in the development pathway and which transcription factors were important and what was the difference between human and mouse to be able to do that. I'll skip that. This is the kind of data that you can see. So I want to just summarize this and say, because we can make novel cell types, we can engineer them for cell therapy, we have isogenic models, we have autologous cells, and we can combine them, we can certainly do a lot more than we could do just even a short time before. Okay? So I want to summarize all of this and say, the regenerative field is very large. There's commercial activity that's taking place in hospitals, and there's a lot of clinic-based activity. There's mostly regulated activity and some unregulated activity. Unregulated activity is in the cells which you can harvest relatively easily from the adults. So it's MSC or bone marrow type cells that you now put into places where you normally wouldn't find them. The field can be divided by the system one is working in. So it's a big field, but you have guys working on neural cells, on cord blood, or on bone marrow. And each subfield has developed differently depending on needs, regulations, etc. In India, we should consider regulations, especially for the clinical-based therapy, and we should clarify regulations for collaborations and approvals of products which have been approved in other countries. And we should have partnerships to test products approved in other countries to get approval here. And we should actively consider what we've looked at about the screening and talks so that we could make sure that that gets done in this country as well. But in everything else, there are people and groups here working on these things as well, both in the regulated and unregulated part of the field. Thank you. So some time left? Huh? Yes, sure.
We have time for questions.
You spoke about the drugs that were toxic in humans after being safe in animals. There must be at least an equal number, if not more, of drugs that have been discarded after animal trials that may not be toxic in humans, so we've lost that benefit. Is anyone working on those drugs?
Yes. So, in fact, there's a big NIH initiative for drug repurposing or drugs which have been abandoned by the pharma industry to rescreen them for use in models that will be there with human models. But I think it's a very important point. And if the ratio of drugs which are rejected is 10 times more than the number of drugs that are approved, so that's a big pool for a lot of effort that's gone into it. So either I was very clear or I was very boring. So
you stunned the audience there. There's one question in the back.
In the industry, how often has the disconnect between behavioral stem cells in a dish versus in vivo affected stem cell-based therapy development?
So with the adult stem cell population, which is the one which has been studied the most, the behavior of cells in a dish has not affected predictions of how they will behave in vivo to a large extent. So nothing that's been a dramatic surprise. It's just been an extent because we don't have a good model. However, with cells where we have to mature them from embryonic stem cells or induced pluripotent cells, right, the behavior of cells in a complex environment is hard to predict from the behavior in a dish. And the rate at which they will differentiate in a dish is different from the rate they differentiate in vivo. So there we don't have good prediction in being able to do that. But with the adult cells, which are short-term in culture, like in bone marrow stem cells, etc., it's been relatively good as a prediction.
So in terms of using the human fetal cell grafts as a potential source of embryonic stem cells, Is it ethically possible to get purified viable cells in terms of enrichment and anything?
Yes, it is possible, but it's difficult, because it takes you have to do some kind of sorting or some kind of selection, and generally cells grow, all adult cells, whether they come from a fetus or they come from young adult or they come from an old, have a lifespan and they undergo senescence, whether they're stem cells or not, which is a different thing from embryonic stem cells or induced pluripotent stem cells. And so it's a lot more complicated,
and it's hard to direct the differentiation in vivo for these cells.
Yes, so you have to push it.
So I had a question regarding these organoids. When you grow them, how do they get shape?
How do you
Or
get?
do you have to give them a particular shape? Like, for example, if you were to culture, say, a heart, it has a very unique shape. So when you grow them, say, once you can vascularize them and whatnot, or do you do, like, for example, the bladder, it grows as a sheet, whereas you'd like them to be spherical or whatever shape they have. So can you get organoids of a particular shape if you just put the cells together, or do you have to direct it to get a shape?
You have to direct it. So there are some self-organizing principles. So the initial part seems to be self-directed. So forming, let's say, involution, or going from between one layer to a second layer, or segregating between cells, these are sort of basic properties of cells, not just stem cells. And so those happen. So let's say for the eye formation that I just showed you with Dr. Anand Swarup's data that was done, the initial part of making the eye is making a ball which segregates from the central nervous system, right? That part happens, and the cavitation happens without any specification. But getting layering happens without specification. But getting the right size, getting the cornea to be in the right place, etc., requires direction and signals, which we have to provide. So the best example of that, the simplest example of what we have to do in culture is when you're making a blood vessel, right? There's a difference between a vein and an artery in terms of the thickness of the muscle. So if you don't provide the right pressure differences, you won't get that. And the diameter of the artery or the vein that you will get, and if you don't provide the right signal in terms of scaffold or something to be able to do it. In this case, normally we put tubes of different thickness on which you grow them, then you won't get the right size to be able to do that. Same thing for a valve, you have to provide a scaffold. For the bladder, we actually gradually inflate a hollow balloon on which you've plated the cells, so that you can get the shape of the bladder and get a hollow organ to be able to do that. And so people then do organoids. They can do tissue printing where they can print cells, but you have to provide some signal after a certain stage, otherwise you won't get that complex.
Another question was, how many cell types do you need? Like, do you need to get just one stem cell type at the beginning to make a bladder, or do you need to provide all the different kinds of
So
stem cells?
here's the rule of thumb, and this is, we are trying to mimic normal development, right? And so if you look at your body, it's normally formed from ectoderm, endoderm, and mesoderm. So all organs have something which comes from ectoderm, endoderm, and mesoderm. So a stem cell for each organ, for example, right, a liver, will provide most of the main cells that are present there. But if you need the vasculature, then you'll have to provide some endothelial cell. And if you need the membranes or the limiting membranes and so on, there'll be some kind of stromal tissue that you'll have to provide. So let's say if I'm making a liver organoid, I need to provide liver stem cells. If I can't provide liver stem cells, I'll have to provide the hepatocyte and the Kupffer cell and the stellate cell, and I'll have to provide some vascular seeding cell and some stromal cell with it to be able to make an organoid. But luckily, there's a lot of self-organization there in terms of being able to do that. And I'll tell you what I mean by that. On average, when you look at any organ or tissue, then within 8 cell diameter, you have a capillary, right? So that's about the diffusion gradient that you can have. So if you mix endothelial cells and liver cells and you let them organize, that's what you'll find. That you'll form these sort of sinusoids which are about that distance. So there's some organizational principle that we don't understand or at least don't know enough to control, and but that's what happens.
Sir, you talked about decellularization and recellularization. So what happens, for example, in a kidney, there are many kinds of cells, like he spoke. So while refilling them with stem cell types, how do you, for example, like first you give some cell type and then how is that process taking place?
So it's very crude right now. So what you do is you just give a mixture of cells back. and you do it through the blood vessels, right? And during this whole process, there's enough damage that the cells actually accumulate as little clumps outside the broken blood vessels, but they're well spread through the whole tissue. And there seem to be signals that are present in the extracellular matrix, which then tell the cells how to organize. And so it's quite amazing how well they organize after that. Meaning, if I cut a histology section, I won't be able to distinguish between those, at least at that resolution.
Is there any process to remove the dead cells from them?
Yeah, there's a wash. So what you're doing is you add something which will kill cells, something like equivalent of ammonia, or something which will kill all the cells. And everybody has some proprietary thing. But then you have to wash all the debris out, and that is very critical, because otherwise the cells won't seed to be able to do that. So earlier the easiest thing was for making blood vessels, and we couldn't get enough from human cadaveric donors, so people found that you could take pig blood vessels, and if you killed off all the cells, then the extracellular matrix didn't cause a huge immune response. And then they found that the big problem there was that when you killed all the cells and they didn't get a huge immune response, then you would get blood clots. So then they said, if we coat it with the human endothelial cells, then that will work. And so that's how it slowly developed. But that was the issue.
Thank you.
Question here, and then last question.
In the drug screening, the recent trend is to use tissue-specific cells as a monolayer. Do you anticipate, just like animal trials, here also you'll have false positive and negative rates, because you don't have the complexity there?
Yeah, so actually that's a very, very important question, and I glossed it over, and I apologize for that. So,
Can you repeat the
okay, sure. So the question here was, yeah, you made all this great point about saying that human cells will be better than rodent cells, right? But how much better? Won't you still get false positive and false negative, right? And what do you expect when you do that? Is that correct? And the answer is yes, you have to be very careful, because here was one thing we learned from pharmacogenomics, okay? And that was that the difference between individuals is as much as difference between a single human and a particular animal, right? So that's the range. So humans are not all the same in that sense. So the way we metabolize drugs, everything else is different. So if I make a stem cell line from one individual, it will be representative of that individual, but it may not be representative of the species. So we may get false positive and false negative. So the trend is to actually make a representative panel, and then do the screen. But we fully expect that we will get false positives and false negatives even there. But we think the number will be much smaller. But to address that issue, what people are proposing is to do what's called a combined trial. So when you're doing a phase 2 or a phase 3 trial, so you're doing 100, 150 patients, that you get cells from them, and so that you can match the adverse events that are happening in a dish at the same time, so that you'll get an answer and a prediction for that.
We'll have a last question in the back.
I had a similar question. Somebody else can ask.
Okay.
Oh.
You have a question?
Can
But she has the mic, so.
How far ahead are we in using stem cells to treat spinal cord injuries? And
to treat spinal cord injuries?
And
So actually, quite a bit has been done, and there are phase 2 trials that are ongoing in spinal cord injury. And there's a difference between the two kinds of spinal cord injuries that people are looking at treating. There's acute, where within the first 7 to 10 days in humans where you can treat a spinal cord injury, and then there's chronic, right? So where you've already had it for more than 3 months and you've got a relatively stable spinal cord injury. And in general, we found that what we can't do is replace the neurons, which take a really long time, right? And that's what's interrupted. But what people found was that if they take a mixture of neurons and astrocytes and put them in the cavity that's formed at the site of spinal cord injury, then what happens is you get a new relay, and that is good enough to get you control of your body. It grows without having to take as much time as it would have taken otherwise to be able to get repair. And that, the data in animals has been really very exciting. And people have been able to now start doing that in humans, and the data is looking quite good in these early studies that have been done.
I think we'll take the last question.
Just a condition of that. So you were talking about the synaptic relays. So how does the synaptic transmission, for example, that occur in the eye, if you transplant the RP? So if you look at the eye, developing the RP sheet is one challenge. Injecting that subretinally is another challenge. But the next step of how do they form the synaptic transmissions, So how does that compare in the eye versus, for example, in the neuronal cells, like in the ALS, for example?
So it's a complicated question, but let me try and answer it the following way. So when people have a stroke, right, you get paralysis, right? And in a lot of people, you recover, and that recovery is 90%, right? Or a lot when that happens. And in some, actually, you don't know that they've had a stroke. That recovery is not because of replacement of cells. That recovery is because the brain can reorganize its synapses and make do with the fewer cells that it has, as long as it has enough, right? So that's what happens, and you don't see anything missing as far as movement and control is there unless you do fine tests. So when we look at replacement of RPE, etc., we think there's enough signal to help retard the death of cells, and the remaining cells can help reorganize their own synapses, because that is a dynamic process that occurs all the way till you're dying, right? I mean, basically, and so that will be sufficient for you to allow you to maintain or even improve your vision without necessarily getting new functional cells there.
Okay. So with that, we have tea outside. Mahendra is going to be, refreshing his stem cells with refreshments outside. But I also wanted to say it's no wonder that Mahendra is perhaps one of the 10 most influential people working in stem cell and regenerative medicine. So, and we are very fortunate to have Mahendra as a collaborative science chair at INSTEM, driving some of the new developments in this area. Thank you very much, Mahendra.
Thank you all for listening.